approaches on axonal cytoskeletal components of neuron. I don't think I am qualified enough to talk more details, so I will let Ishaq to carry on from here.
Thanks, moderator, and thanks, Satyajit, for inviting me in today's session. It's an honor to talk.
Just a second, Ishaq. Do you have a tool in your team? Because I'm just logging in from my Bangladesh, so I cannot actually record this session. So if you have any recording tool, or if anyone else have recording tool, you can help us.
Let me see who is the host.
Sorry for that.
Okay, I have to be
Actually,
host.
I don't see anyone.
It's showing me I have to
Yeah,
be
so
the
I
host.
actually attended. Yeah, so I was actually the host, but somehow I cannot log in through my Northwestern ID. So
okay.
this
Can
is
Moinuddin
the
Shubho do
problem.
it?
It's so claiming host. I will just say enter the host key to claim host role. Host key is 6 to 10 digit number.
Okay. Give me 1 or 2 minutes. I think I can fix it. Okay, sorry for the problem.
Satyajit, is everything fine?
I think hey ওনার নর্থ ওয়েস্টার্ন লগ ইন আইডি দিয়ে ওনার লগ ইন করতে হবে হোস্ট রিলেটেড সমস্যাটা রিজলভ করার জন্য।
আচ্ছা,
let me see if I can log in from my account and get the account.
Okay, sure.
সরি অ্যাকচুয়ালি আমরা লগ ইন করতে পারতেছে না, তো আই থিঙ্ক উই নিড সাম অল্টারনেটিভ। প্রত্যয় তুমি কি ইয়াটা ইনস্টল করতে পারছো?
আপনার হচ্ছে স্ক্রিন রেকর্ড করছে, ও কেবলই আমাকে হচ্ছে কনফার্ম করলো যে আপনি স্ক্রিন রেকর্ড করতে।
তো এখন কি স্ক্রিন রেকর্ড হচ্ছে?
স্যার আমি হচ্ছে স্ক্রিন রেকর্ড করছি, বাট আমার এখানে একটু ইলেকট্রিসিটি প্রবলেম দেয়, সো আরো অল্টারনেট কেউ। ওকে স্যার, আমরা দুইজন করছি, সো সমস্যা নাই, আমরা ওবিএস দিয়ে করছি।
I think then we can go ahead. So, Isaac, you can go ahead. Sorry for the interrupt and problem.
All right. Thank you so much for inviting me in this seminar. And the title for today's webinar that I have set is kind of generalized because as far as Satyajit told me that it's just about sharing ideas about how to solve this kind of problems using this kind of tools, meaning how to solve biophysical problems with computational mechanics. That's why I'm not going into so much detail of the mathematics and everything, but I'd like to give an idea to the general audience on how to use these computational mechanics kind of tools and specifically computational tools to solve biomolecular systems or biophysical problems, which are very relevant to real life research problems. My specific approach would be to kind of taking an attempt to visualize such kind of problem. So let's go ahead. The slide is changing, right?
Yeah.
Okay, so as Shubho has introduced me, my work was on computational characterization of axonal cytoskeletal components. It's full of so many terminologies of multidisciplinary expertise, but let me break it down. In brain, we know that there are more than 20 billions of neurons, and each neuron contains an axon. A rod-shaped axon. You know what we have read in probably high school biology books, that a neuron contains a soma, that is a cell body, some dendrites which look like tree branches, and an axon that creates the link between one neuron and another neuron. So that axon itself, it consists of some subaxonal components. Those are essentially proteins. And these proteins are structurally different. For example, as you can see in this table, that protein can be ordered, partially disordered, and intrinsically disordered, meaning that they can have a very fixed or conserved structure, or a part of the structure can be disordered, meaning they can be very flexible. They don't have any specific structure. And sometimes the entire structure can be disordered. What do I mean by that? You can imagine an ordered structure as a dry packet of instant noodle. It's very hard. It has a definite shape. But when it's disordered, you can imagine as boiling spaghetti in a bowl. So that it's very flexible and it can move around. And from these images, you can see that proteins can be of different structures. And now, when it comes to axon, there are different types of subaxonal components. Namely, there are 4 major components: microtubules, microfilaments, neurofilaments, and tau protein. You can see that microtubules and microfilaments are very structured. We don't have to worry about their structure. Neurofilament has a disordered portion in its structure. Some portion is structured and the rest is disordered. And the tau protein, which is actually a cross-link between different microtubule bundles, this is intrinsically disordered protein. Now, if we want to solve a biophysical problem in terms of a brain problem, we have to find out what is a critical problem in terms of brain injury or brain neurodegeneration. And when we try to analyze those problems, we have to know this kind of definitions. What are in those subaxonal components, and what are their structures and how they behave? And that's why when we try to solve a problem relating to brain injury, we have to know that there is a length scale hierarchy in brain. For example, if you hit someone on the head, you can immediately see, or may not see, depending on the extent of the injury, that that person is injured in the head. You are talking about macro scale. But if you know about the anisotropy and length scale of the brain mechanical properties of the components of brain, you can see that the propagation of injury and extent of injury is different at different length scale. For example, in macro scale, the injury will be different. At tissue scale, the injury will be different. And when you go into very, very small length scale, for example, in nanometer, you are in the axon level, and you can see the microtubule, neurofilament, and tau protein. Those will also behave differently according to the extent of the damage. And that's why I'm actually at the very bottom of this hierarchy at nanometer level. So during my PhD work, I have actually analyzed how they mechanically deform this type of microtubule, neurofilament, and tau protein. How do they deform when somebody is injured in the head? So you are taking the injury conditions from the macro scale, and you are trying to imitate computationally by using different tools, atomistic simulation tools, what happens at the cytoskeleton levels. And when you can determine the mechanical properties of the cytoskeletal components, you can scale up, you can apply those properties to model the single axon, and then can scale up even more. You can go to the network level and then tract level. And when you are scaling up, at some time, you are no longer in the atomistic level. You will be in the continuum level, and you will be doing a multiscale kind of modeling. Here is like another visualization. When there is a head injury, there will be a tissue level injury, but it will be different because tissue level injury will be characterized by how the acceleration and deceleration happens due to the macro scale injury. And due to that, many things happen at the neuron level. For example, there are so many interactions between the subaxonal components, and those interactions will be hampered when someone is injured. For example, there will be some biochemical interactions that is triggered by the injury that will happen. Some post-translational modifications will happen. For example, the structure will be altered in terms of interaction. Some interactions will be hampered so that the molecular integrity will be hampered. So you can see that even a macro scale event can significantly affect neuron level phenomenon. And that's why when we try to solve a brain injury problem, we have to know the major components of the axons and their mechanical structure, their interaction, and associated biophysical and biochemical components. We're talking about biophysical and biochemical components because at the end of the day, these are materials, but these are also biological components. They have not only properties as a material, they also have biological functions, and they interact with the environment according to the physical rules. And these are highly altered by the chemical phenomena according to their circumstances. That's why there are so many things and so many parameters associated with them. Whenever you are seeing a mechanical deformation in a microtubule or tau protein, you also have to consider what is their surroundings, meaning what is their physics, and also what kind of chemical phenomenon are affected by this kind of injury, meaning you are also incorporating chemistry here. That's why when it comes to a biophysical problem, there are mechanical, chemical, and physical components you are talking about. Now, let's say we have a simple hypothesis that is, you are a brain scientist. You are trying to address a research problem regarding brain disease. Say you want to address a brain disease. You want to cure a brain disease. And you want to use computational mechanics to solve a brain disease problem. That is, you want to find out how brain disease propagates or how we can solve by using computational mechanics. Now, what kind of specific research questions those can be? Let's say a person is injured in the head. We can ask what happens at the nanoscale? What kind of interactions are being hindered? And what kind of structural changes are occurring? And how can you determine the damage threshold or injury threshold? And can we actually successfully or meaningfully simulate the deformation of the individual components? Can we scale up these problems to continuum level? And can we, at the end of the day, we want to cure the damage. So what can we do to stop the damage propagation? How can we heal the brain? How can we trigger the healing mechanism of the brain? So there are so many questions you can ask. Some of these are already answered. You have to find which questions are not answered so that you can be the connecting dot to solve possibly a high-scale research question 5 years down the line. And let's say we are trying to address a very specific brain disease, specific kind of brain disease like Alzheimer's disease or chronic traumatic encephalopathy. We can see the visualization, what happens at macro scale when a normal brain is affected by those kind of diseases. Just a disclaimer, this kind of disorders or diseases can take place when due to many different reasons, not only injury, but it can also happen due to aging. That is, when a person becomes old, the mechanism of brain degradation starts to take place, and they lose their memory due to volume loss in several brain spaces, and that sort of stuff. But a lot of the time, this is also associated with brain injury. For example, an Alzheimer's disease case that can be worsened in an injured person. So there's a difference between a non-injured Alzheimer's disease person and an injured Alzheimer's disease person, and so and similarly that goes for CTE also. And how do we know that this kind of injury is propagating in these diseased brains? There are several kinds of biomarkers, like loss of brain volume or loss of gyri and sulci, and losing of brain integration, that sort of things. And you can also characterize this kind of injuries in nanoscale. For example, you will see that several proteins related to disease, these are deposited in different parts of brains, which will tell you as a doctor or a neuroscientist that this brain is no longer healthy, there is something happening. For example, let's say we're taking a very specific example. This is entirely hypothetical. I'm using the most known proteins so that many people can relate. Let's say we have a hypothesis that when someone is injured, a pathology or a disease propagates. Why? Because when someone is injured, a very specific injury-related protein, amyloid beta, that aggregates. Aggregates means that it starts to deposit in different parts of the brain, so that if it goes if it goes beyond a threshold value, we can say that there is a fibrillar aggregation, amyloid beta is happening, and it's forming beta sheets. And when this is happening, we can say that the brain is getting affected. It's getting a disease. And we have several hypotheses on how to remedy this situation. We can propose that possibly if we get this amyloid beta to interact with something called X, another protein, possibly this beta formation will break, because in most of the cases in these biological components, their functionality are interaction dependent. When they interact with a specific kind of protein or an enzyme or something like that, something kind of a trigger, their functionalities or behaviors will change. That is, these will act as a switch. So we can hypothesize, let's say based on the existing literature, that if we get these amyloid beta sheets to interact with glutamate, I'm using glutamate because it's the most abundant protein in the brain. Let's say if it interacts with amyloid beta, these beta sheet formations will break. And when beta sheet formations will break, the disease is no longer there. The cause of the disease is remedied. So it goes to the healthy form of brain. So that's our working hypothesis for now. Let's say that someone is injured and this person is taken to the hospital. You are seeing that in different parts of the brain, he has amyloid beta sheet formation, and you are trying to see if we try to interact amyloid beta with glutamate, this beta sheet formation will be inhibited, and that's how he will be cured. So that is a very large scale problem, but the key step is to see the amyloid beta glutamate interaction. You don't know yet what will happen if these two will interact. So that's why you have to simulate. Let's say you have several computational tools at your disposal, so that you want to simulate these interaction, amyloid beta and glutamate interaction. So the first step to see this interaction is molecular docking. Why molecular docking? First of all, it's very small. You are in nanoscale, and these two are protein, so you can actually use protein-protein interaction. And there are several servers that you can use. There are several manual algorithms or codes that you can use to simulate their interactions. Let's say here the big protein is the amyloid beta, and the small protein is the glutamate. For this purpose, we can say that this is a ligand which will come and interact with the amyloid beta. We want to see how it interacts with amyloid beta structure. So let's say we are going to some docking servers. Some popular docking servers are PatchDock and FireDock or ClassPro. You can also use Haddock, or you can use GramX. You can use Rosetta, and sometimes they allow you some flexibility in terms of this kind of designing on how you want to see them interact. Sometimes you can determine their binding affinity, binding energies, and also like favorable docking positions. So you can see that when two proteins interact, they don't interact by random. You cannot like get to interact them as you wish. They will interact in terms of physical and chemical favorability, or more generally energy favorability. You can see that in a protein structure, it's actually an amino acid sequence. Some of these are acidic, some of these are not acidic, some of these are electrostatically positive or negative. They have charged portions, And there are hydrophobicity and hydrophilic interactions that you have to take into consideration. So when you get two proteins close towards one another to interact, they will interact following different criteria. For example, a hydrophobic region will interact in a way that is different from a portion that is not hydrophobic, or an electrostatically positive portion will attract an electronegative portion of another protein, and that's how they will interact. Sometimes they will also follow induced fit mechanism, like a lock and key kind of mechanism. You can see that you cannot insert a key into a lock in any way you wish. You have to align the key with the lock, and that's how you get to interact a protein with another protein. So these kind of servers actually use different algorithms to facilitate you using these kind of principles of protein-protein interaction. Now, if you submit the structures of amyloid beta and glutamate into these servers or your software like Rosetta, you will get some posed structures, like a combined structures, amyloid beta glutamate combined structures. How many? 10, 100, or 1,000. And you can refine them based on energy favorability. For example, in PatchDoc, when you submit, you will get like 1,000 structures that can energetically happen, but you refine them by sending them to FireDoc and take only top 10, because in terms of computational simulation, you want to minimize the use of your usage of resources, and you want to be reasonable in terms of simulation. So let's say you are proceeding with 10 combined structures, that is some docked poses, and then you go to molecular dynamics. You want to see their dynamic behavior. This kind of docked poses, initial poses, these are your initial t equals 0 position to see their interaction. And then in a molecular dynamics simulation environment, you can see their interaction, whether this system is stable, whether these are interacting, how they're interacting. Is this interaction very rigid? Are they attracting each other very well? Or is it momentary? Like, is amyloid beta interacting with glutamate for just a few picoseconds, and then glutamate is going away? So you will get an indication that they do not like each other. And other kind of energetical change, like let's say the entire system collapses when you are running molecular dynamics, or you are seeing a very specific kind of attraction or change of interaction. That sort of things are your observables when you are running a molecular dynamics simulations. So let's say you are getting two different observations, very specific. I'm getting very specific to show you like how we would like to address these kind of research questions. Let's say you have a control. In all kind of experiments or computations, there is always a control, meaning that there is nothing here, only your default system, so that you can compare it with other non-control observations. Let's say you are running a molecular dynamics simulations or amyloid beta only. Okay, so you have run it for 100 nanoseconds, and you are seeing RMSD value, that is root mean squared deviation. In simpler terms, it's like how much did the protein move. It actually shows you a quantification of the stability of the system. Let's say in only amyloid beta system, there is no glutamate there, only amyloid beta system, you are seeing that it's very unstable. Amyloid beta is very unstable. It's moving 20 angstrom here and there. So you can see that this system is very unstable. But when you added glutamate with your system, you have 10 docked poses of amyloid beta and glutamate, and you have run it for 100 nanoseconds, and you can see that it has suddenly become very stable. It's not moving at all. It's just 1 angstrom. The RMSD value is 1 angstrom. It's meaning that this, when it's combined with glutamate, it's become very stable. And you have actually applied many other scientific principles, like you are doing it repeatedly so that you can make sure that it's not just by chance, but you can repeat and reproduce the result. You are running it for sufficient time so that you are in a reasonable range in terms of time scale. And you are also checking the molecular dynamics simulation parameters, like whether your force field is valid for this kind of systems. What is force field? I will come into later portion of this presentation. So let's say from these observations, you can actually take a decision. Let's say that first you hypothesize that if we get amyloid beta to interact with glutamate, this will be like breaking up amyloid beta. Instead, it's actually stabilizing the amyloid beta system. So you can say that your initial hypothesis was wrong. You were very wrong your initial hypothesis. So you should actually remove glutamate from your system so that the amyloid beta system cannot propagate. Its fibrillar conformation cannot propagate. So that's a very simplified way to see or interpret the results of molecular dynamics. And when it comes to molecular dynamics, I should clarify some of the terminologies related to molecular dynamics. There are several platforms in which run this kind of simulations. For example, Yasara, LAMMPS, NAMD, or GROMACS. These are packages. When we say to someone that I'm running molecular dynamics, essentially we're implying that we're using one of these kind of packages. There are several others. I'm just mentioning here the most popular ones. And what are inside those packages? There are possibly graphical user interface, or there may not be there. There might be only command line prompt or coding platform. And there are essentially force fields. And what do I mean by force fields? I'm coming into that. That's a very crucial part of molecular dynamics. And when I'm talking about molecular dynamics packages, what is inside there? In molecular dynamics simulation, this is actually a screenshot from a notepad file that I generally use when I mentor molecular dynamics simulation, or when I teach this kind of things. I prefer to use notepad because of the simplicity of it. When it comes to different packages for let's say Yasara, LAMMPS, or NAMD, there are different things. For example, in LAMMPS, you have a structure file, the protein that you want to simulate. You have to make a structure file for that, and you have to make an input file in terms of commands. In that input file, you have some kind of commands regarding this structure, what you want to do with that structure. For example, for this amyloid beta and glutamate structure, you will possibly be using some kind of interactions. So you want to equilibrate it for a long time, for example 100 nanosecond. And as an output, you get a dump file where you can see the trajectories and everything. So similarly, in other packages, you have these three things: structure, commands, and output. When you are using an NAMD file, when you are using the NAMD package, in structure, you are using a PDB file. In terms of commands, you are using a configuration, and as output, you will see trajectory, meaning the velocities and coordinates and other things that you want to see as an output. When it comes to Yasara, it's also kind of the same. So in the input file, you generally set this kind of things: the structure information, the simulation box size, the environment. Environment setting is very crucial, like how you would like the protein to interact with the environment, and what's your time step and what's the duration. So that kind of things you actually set in a molecular dynamics package, and then you run your simulation, and then you analyze your data from the dump file or the trajectory file, whatever the output is. And I have repeatedly said about the force fields. When it comes to a protein structure, you have to actually define two different things. One is that what is in that structure. A protein structure, in a general sense, it's a sequence of amino acids, but if you break it down, it's actually a combination of atoms, let's say 100 or 1000 atoms. And how does it become a structure? By the interaction between the atoms. For example, let's say if we consider a body, our head is connected to the body, connected to the rest of the body through our neck, and it's only a very fixed orientation. Our hand is connected to our body with a different orientation. It has an elbow that has a ball kind of joint. We have shoulder that interacts differently. So similarly, in a structure, a protein structure, let's say in one amino acid, there are 20 atoms, let's say. And these 20 atoms are actually connected to each other with different orientations. They have bonds, they have angles with each other, they have out of plane angles, that is dihedral angles with them. So that is their bonded parameters. There are also non-bonded parameters. They have also some other properties, like where they're situated. Are they close enough to form an attraction or repulsion? And or what charge do they have, positive, negative, or neutral? So in a structure, a protein structure, when you are preparing it for an MD simulation, you have to actually define these kind of things in terms of a functional form. At that functional form of energy, that is called a force field. And it's where does this force field come from? How do I know that I can define the bonds and angles and dihedrals and the Yim-Propers, Urey-Bradley, non-bonded interactions like electrostatic and van der Waals interactions like this? How do I know that this is correct? This actually comes from experimental parameterization. These come from empirical observations, and when we run an experiment of this kind of proteins, we can actually extract their bond angle dihedral information or this kind of parameters, and we can actually parameterize them in terms of computational force fields. And so that when we try to simulate a behavior, a mechanical behavior or biophysical behavior or chemical behavior through our force fields, we try to capture their energy properties in terms of their bonds and angles and dihedrals and non-bonded interactions. We actually define them in terms of empirical observations. And that's how the force fields are developed. Very popular examples of protein system simulations are CHARM, AMBER, GROMOS, or Martini. You can look into those functional forms of these force fields, and you can see that there are similarities, but some tweaks and turns in terms of the functional forms, because the research is ongoing on how to efficiently and realistically capture their behavior so that it will be similar in computation as we see in experiments. So at the end of the day, when we are trying to simulate these kind of biomechanical or chemomechanical problem, we end up with some questions like when it is an injured brain, we know that how the injury is, what kind of strain rate or stress strain that occurs at the subaxonal components, or like amyloid beta, and how does it affect the amyloid beta glutamate interaction. For example, if you want to see how it is affecting amyloid beta glutamate interaction, how do we quantify this? We will compare, possibly we'll compare two different amyloid beta glutamate interaction systems. In one system, there will be no injury, and in another system, there will be injury. For example, we will apply a velocity field to this interaction to see if any kind of mechanical injury in these nanoscale length affects this kind of interaction, and we can relate it to our observation. Like amyloid beta glutamate interaction does this, it stabilizes the system, and the injury does it better to them, or whether it worsens it. So that's how we can quantify the deformation by using computational mechanics principles. You can actually apply a force to a biomolecular system by using the classical mechanics principles, and that's how the computational mechanics comes into play when to determine the mechanical properties or to solve a biomechanical problem. Here I have used some stock photos of steered molecular dynamics. It's showing that one person is actually pulling away a protein from another protein, and the others are holding it. This is kind of happens when you are trying to simulate a protein-protein interaction. We see whether a two-protein system is stable, and then when it's stable enough, we try to apply a deformation to the field so that we can know that what is their separation stretch, are these interactions strong enough, or that sort of things. And then when we get the insight at the nanoscale level, we can actually scale up as I have said at the beginning of my presentation, that when we scale up enough, it no longer stays at an atomistic problem. It becomes a continuum problem. For example, we get the insight of the mechanical properties of the subaxonal components, then you can scale up. We can apply it to model the whole axon, and then we get when we get the property of the whole axon, we can scale up again. We can model the axon network, and when we get the property of the axon model again, we can scale up, and then and slowly but surely we can, considering the computational resource and other constraints, we can actually scale up to the continuum problem. And then we can essentially characterize a head model, a head injury problem, where you have actually realistically defined a very high definition continuum model of a brain. So that's how this problem goes down the line, and this is called a bottom-up approach. You are starting at the very nanometer level, and then you are scaling up to solve a head level problem. So that's how, and also you are like getting novel insights on how brain injury or brain degradation is related to this kind of subaxonal level interactions. So and what are our observables? Like I'm talking repeatedly about properties, but what do we actually see when we apply any deformation to these kind of small scale problems? We can firstly, I have said that it's a very multidisciplinary problem. There are biological, mechanical, and chemical components. So we see the chemical stability, and in terms of mechanical property, we can see force displacement or stress strain. And also we can say see strain rate dependence when it comes to injury in a biomolecular system. Sometimes there is strain rate dependence. If you pull a protein from another protein at very slowly, it may show remarkable resilience. But if you pull very fast, it may fail it may also fail very fast. And also we can see separation criteria and that sort of things. In terms of stability, sometimes we equilibrate it for a very long time, and we try to see whether we see some interesting interactions, or whether is this RMSD values is zigzagging or not, and that sort of things. And the simulation box essentially contains solvating liquid, so we see also the interaction of the protein with the water, which quantifies the stability. So this is so at the end of the day, we go back to the problem we started with. In the injury, we start with a hypothesis that possibly this kind of injury biomarker called amyloid beta, if we get to interact with it, if we get to see an interaction with amyloid beta with glutamate, possibly amyloid beta will break. In our case, it didn't happen. It happened totally the opposite thing. It actually stabilized the amyloid beta. So the first hypothesis failed. We will proceed with another hypothesis. Possibly we'll test another protein to interact with amyloid beta to see what can actually break it down so that we can restore or heal our brain. So that's how. So this is one aspect of seeing a biophysical problem. In here, we have actually seen the interaction criteria. There can be other approaches to this kind of problem, like mechanical property. What is the mechanical property of amyloid beta? Some people might work on it. What is the damage threshold? What kind of mechanical deformation we have to apply to damage it physically? And what is the mechanism of it in terms of chemical interaction? We have actually captured it here. What is the damage mechanism in terms of chemical interaction? And also sometimes the problems are actually in the methodology. Sometimes you can see the problem as an attempt to validate your force field. Whether you are designing a force field and it's not working realistically for this kind of system, so you can actually tweak some parameters in your force field, and that's how you can develop and come up with a modified or entirely new force field. So there are so many scopes of working here. You can actually see this problem as a mechanical engineer, and also you can see the problem as a structural biologist. You can see this problem as a force field developer. So you can see that it's a very much multidisciplinary problem to say, and that's how we actually address this kind of problems. And down the line, 5 to 10 years, there is always a large objective in mind. Just to simulate the system is not our objective. It's a transition or a step to the ultimate goal, that is curing brain disease or addressing a biophysical problem, so to say. So I hope that without going into any mathematical or rigorous formulation, I think I have been able to show you how to approach real life research problem and how to incorporate your atomistic simulation tools and how to scientifically interpret your results. I hope I have been clear in terms of terminologies and explanation and visualization. Thank you.
Thank you, Sankai, for the detailed presentation. We will start taking questions from the audience now. Anyone have any questions? You can type it in or just unmute yourself and ask away. Is there anyone?
আমার একটা প্রশ্ন ছিল।
Go ahead.
আমি বাংলাতে আগে করছি, ইংলিশে বোঝানো একটু আর কি ডিফিকাল্ট হবে আমার জন্য।
Sure.
আচ্ছা, এখানে ভাইয়া যে উদাহরণটা আর কি দিলেন সেটা হচ্ছে দুইটা প্রোটিনের মলিকুলার ডকিং এর মাধ্যমে তাদের ইন্টারঅ্যাকশনটা স্টেবল হবে অথবা আনস্টেবল হবে এটা, এবং দেখলাম যে এখানে পসিবল হচ্ছে 1000টা কম্বিনেশন, ওইখান থেকে গ্রাজুয়ালি কমাতে 100টা, তারপরে 10টা, তারপরে যে স্টেবল কন্ডিশন যেগুলো আর কি ছিল ওগুলো থেকে হচ্ছে আমরা সিমুলেশন আর কি করেছিলাম। এখন এই জিনিসটা যখন আমরা রিয়েল লাইফে অ্যাপ্লাই করব তখন জিনিসটা একটু রিস্কি হয়ে যায় কিনা, কারণ হচ্ছে আমরা কি আসলে পসিবলি জানতে পারছি যে আমরা যখন আর কি গ্লুটামেটটা যখন এটার সাথে বন্ডিং করাবো তখন কোন মলিকুলার ডকিং পজিশনে এটা আর কি ইন্টারঅ্যাক্ট করছে? এখানে কি কোনো মানে এখানে যদি অন্য কোনো একটা মলিকুলার ডকিং পজিশনে যদি ইন্টারঅ্যাক্ট করে সেক্ষেত্রে জিনিসটা তো মানে আমরা ধরা যাক দেখলাম যে একটা কন্ডিশনের জন্য স্টেবল হলো, কিন্তু রিয়েল লাইফে যখন অ্যাপ্লাই করলাম তখন দেখা গেল যে আর একটা পজিশনে ইন্টারাকশন হলো এবং জিনিসটা আনস্টেবল হয়ে গেল। এরকম কোনো পসিবিলিটি থাকে কিনা, কারণ আমরা মানে এর সাথে প্রবাবিলিটির কোনো সম্পর্ক আছে কিনা এটা জানতে চাচ্ছিলাম।
আচ্ছা, এটা খুবই গুড কোয়েশ্চেন। কারণ হচ্ছে আমি আসলে যেভাবে প্রবলেমটাকে উপস্থাপন করলাম সেটা হচ্ছে লাইক কাইন্ড অফ চেরি পিকিং। ইউ আর taking a problem and you are getting a solution and then proceeding with it. রিয়েল লাইফে এটা কিভাবে ঘটে, এটা অ্যাকচুয়ালি আমরা বলতে পারি একটা ড্রাগ ডেভেলপমেন্টের ভেরি ইনিশিয়াল স্টেজ। ঠিক আছে, আমরা মনে করি যে আমরা নিউরোডিজেনারেশনকে অ্যাড্রেস করার জন্য আমরা হচ্ছে শুরুতে এই কম্পিউটেশনাল স্টাডিটা করলাম, যে আমরা একটা মলিকুলার ডকিং করলাম, অ্যামাইলয়েড বেটার সাথে গ্লুটামেটের একটা ইন্টারাকশন দেখলাম এবং পসিবল 10টা পেলাম। ঠিক আছে, এখন কোশ্চেনটা খুবই স্বাভাবিক আসতে পারে যে আসলে 10টাই যে হবে এরকম তো আসলে কথা নেই, এর চেয়ে অনেক রকম ভাবেও অনেক রকম ভিন্ন ভাবেও হতে পারে। সেটা আমরা কেন এই পসিবিলিটিটা আমরা বাদ দিয়ে চলে যাচ্ছি কিনা, এটা রিস্কি হচ্ছে কিনা ইন টার্মস অফ ইন্ডাস্ট্রি পার্সপেক্টিভ। সো ইট ক্যান হ্যাপেন। সো এই প্রবলেমগুলো অ্যাড্রেস করার জন্য যেটা করা হয় সেটা হচ্ছে বেশ কিছু ক্রস চেক করা হয়। একটা হচ্ছে যে আমরা যেই সার্ভারের মাধ্যমে এই ইন্টারাকশনটা করলাম সেই সার্ভারটা আসলে এই টাইপের সিস্টেমের জন্য ভ্যালিড কিনা সেটা অবশ্যই আমাদেরকে লিটারেচার থেকে ভেরিফাই করতে হয়। এবং আমরা যেই ইন্টারাকশন গুলো পেলাম সেই ইন্টারাকশন গুলোর থেকে আমাদের নিতে হয় যত বেশি সম্ভব স্যাম্পল সাইজ, যত বড় সম্ভব স্যাম্পল সাইজ নিতে হয়। যেমন আমি এখানে বললাম যে উদাহরণ হিসেবে একটা স্যাম্পল সাইজ নিয়ে মানে একটা রেজাল্ট নিয়ে আমি শুরু করে দিলাম ইনিশিয়াল নিয়ে, সেটা হতে পারে একটা টেস্ট কেস, বাট ইন জেনারেল আমরা হচ্ছে অনেকগুলো ইন্টারাকশন নিই। ঠিক আছে, এবং তাদের কিন্তু আমরা যখন ইন্টারঅ্যাক্ট করছি তখন সেটার কিছু ওয়েইং প্যারামিটারও কিন্তু থাকে। যেমন আমি হচ্ছে বেশ কিছু জিনিস এখানে স্কিপ করেছি সময়ের স্বার্থে, যে প্রত্যেকটা ডকিং পোজ কামস উইথ আ স্কোর, কেমন কনফিডেন্স স্কোর সহকারে আসে কিন্তু, যে এটা কতটা রিলায়েবল ইন টার্মস অফ এনার্জেটিক্স। এবং আমরা কিন্তু এখানেই জিনিসটাকে ছেড়ে দিচ্ছি না, যে আমরা কিন্তু বলে দিচ্ছি না যে এখানেই এভাবেই ওর ইন্টারাকশন হবে, আমরা ইন্টারাকশনটাকে টেস্ট করছি মলিকুলার ডিনামিক্স এর মাধ্যমে। সো আনসারটা হচ্ছে এই যে এটা অ্যাড্রেস করা হয় বড় স্যাম্পল সাইজ ইউজ করার মাধ্যমে, হয়তো 10টার বদলে আমরা 100টা নিয়ে হয়তো টেস্ট করব, এবং প্রত্যেকটা হবে কি এনার্জেটিকলি ফেভারেবল পোজ। তার মানে হচ্ছে যতগুলো পোজ সম্ভব, পোজ আসলে অনেক সম্ভব, 1000-10,000 সম্ভব, বাট in terms of computational resource we have to use 10 to 100। ঠিক আছে, এবং তাদের স্টেবিলিটিটা খুবই রিগোরাসলি টেস্ট করা হয়। সাধারণত ইন জেনারেল রুল অফ থাম হচ্ছে 100 ন্যানোসেকেন্ড বা এরকম করা, অন্যান্য সিস্টেমের ক্ষেত্রে কম বেশি হতে পারে, বাট এটার আসলে ক্যাপাবিলিটিটা দিনে দিনে বাড়ছে এবং বর্তমানে 1 থেকে 2 মাইক্রোসেকেন্ড পর্যন্ত করা হয়। সাধারণত ততক্ষণ পর্যন্তই করা হয় যতক্ষণ পর্যন্ত করলে আমরা এটার কেমিক্যাল এবং বায়োফিজিক্যাল বিহেভিয়ার সম্পর্কে জানতে পারি আর কি। প্রত্যেকটা স্ট্রাকচারের সাথেই একটা অ্যাসোসিয়েটেড টাইম স্কেল থাকে যে এটার স্টেবল স্ট্রাকচারে আসতে কতটুকু সময় লাগতে পারে। তো সেভাবেই আর কি করা হয়, এবং এই ব্যাপারটা যখন আমরা ভ্যালিডেট করে ফেলতে পারি এনার্জেটিকলি তখন কিন্তু শুরুতেই এটা মানে রিস্কের কথাটা যখন আসলো, এবং এটা যেহেতু ড্রাগ ডেভেলপমেন্টের সাথে রিলেটেড, সো এটা শুরুতেই কিন্তু মানে এফডিএ অ্যাপ্রুভ করে দিবে না। শুরুতে যেটা হবে সেটা ইনভিট্রোতে যাবে, ইনভিট্রো এক্সাম্পলে যাবে, ইনভিট্রো টেস্টে যাবে। সেখানেও হচ্ছে একটা রিগোরাস স্টেপের মধ্যে দিয়ে যাবে যে এই টাইপের ইন্টারাকশন আসলেই ব্রেইনে ঘটে কিনা। আমরা যে কম্পিউটেশনাল সিমুলেশনে দেখলাম সেটা আমরা একটা ইনভিট্রো সিস্টেমে সিমুলেট করতে পারি কিনা সেটা দেখা হবে। তারপর যখন ইনভিট্রোতে হবে তখন তারা ইন ভিভো টেস্টে যাবে, ঠিক আছে। ইন ভিভো টেস্টে মানে হচ্ছে একেবারেই ফিজিক্যাল যে এক্সপেরিমেন্ট সেটার মধ্যে যাবে। ফিজিক্যাল এক্সপেরিমেন্ট বলতে হয়তোবা শুরুতে অ্যানিম্যাল মডেলের উপরে তারা এটা টেস্ট করে দেখবে, সেখানেও যদি তারা দেখে যে এখানে আমরা একই রকম রেজাল্ট পাচ্ছি, তখন তারা যাবে হচ্ছে ক্লিনিক্যাল টেস্টে, অর্থাৎ তারা হচ্ছে তখন এটাকে ড্রাগ আকারেই একটা প্রোটোটাইপ ডেভেলপ করবে এবং সেটাকে ছোট স্কেলে মানুষের উপর অ্যাপ্লাই করে দেখবে যে এই ধরনের ইন্টারাকশনই আসলে পাচ্ছে কিনা, এবং তারপর সেটাতেও যদি একই রকম রেজাল্ট পাওয়া যায় তখন সে অ্যাপ্রুভাল হবে। সো there are so many check marks here that you have to pass through. কাজেই রিস্কের যে ব্যাপারটা, এটা অ্যাকচুয়ালি ভেরি ফার্স্ট স্টেজ অফ মলিকুলার ডকিং এই আসলে শুড বি টেকেন কেয়ার অফ। বাট তারপরেও হচ্ছে প্রত্যেকটা স্টেজেই কিন্তু এই জিনিসগুলো ভেরিফাই করে যেতে হবে যে এনার্জেটিকলি ফেভারেবল কিনা, এবং আমরা কোন রকম আনফেভারেবল পোজ বা আনফেভারেবল ইন্টারাকশন নিয়ে পাস করে যাওয়াটা আসলে ভেরি আনলাইকলি। আনসারটা কি পাওয়া গেল?
জি ভাই, বুঝতে পেরেছি।
Great. Yeah, we're gonna take a couple of more questions. I guess the time was up to 12, right? আমরা আর একটা দুইটা প্রশ্ন নিতে পারি if anyone wants to ask away. দাদা, আপনি মনে হয় এখনো ফোন দিয়ে আছেন, সত্যজিৎ, আপনি চাইলে কিছু বলতে পারেন।
So I have a quick question. So you are using dynamic simulation for the kind of materials. My question is, are you using all atomic model or a kind of screen molecular dynamics?
I didn't get that. It's like
You are breaking off. Could you?
Oh, sorry. So my connection is very. Maybe I can just type in and you can.
That'll be convenient, I guess.
Okay, so I'm typing in the tab. By the time you can also ask. Yeah.
If anyone else wants to ask any more questions, please go ahead or type in. I want to আমার research is a completely different field, so it might be a naive question, but I wanted to know, so the modeling you're doing right now, does it start from continuum level and then goes to atomic scale, or is it the other way around?
It's the other way around. আমি একটা সময় বললাম না যে এটা আসলে বটম আপ অ্যাপ্রোচ কাইন্ড অফ থিং। So you can search for one term. It's called atomistic based continuum modeling. So you start at the very bottom, like sub-axonal level modeling, and then you get the mechanical properties of those sub-axonal components, and then you can apply those properties to a kind of bigger model like whole axon, which contains all of these. Now when you keep scaling up like this, you get a very big model with so much details, because if you start at the continuum scale, what happens is that you just make a head model with possibly a million elements, let's say. আমরা একটা ব্রেইনের এফইএম মডেল নিলাম, সেটাকে আমরা, এটা করা যেতেই পারে, আমাদের হচ্ছে এমআরআই ইমেজ অনেকগুলো নিলাম, সেটাকে স্ট্যাক করে একটা 3D মডেলে আমরা রূপান্তর করলাম, সেটাকে আমরা মেশে কনভার্ট করে আমরা এফইএম মডেলিং শুরু করলাম। কিন্তু এটাতে প্রবলেম হবে কি, যে এটা আসলে ছোট কম্পোনেন্ট গুলোর যে মেকানিক্যাল সাপোর্টটা দিচ্ছে সেটা এখানে ক্যাপচার করবে না। এফইএম মডেলগুলোর একটা বড় ইয়ে হচ্ছে কি, লিমিটেশন হচ্ছে কি, যে এখানে দেখা যায় যে শুধুমাত্র 2টা কি 3টা মেজর কম্পোনেন্টের প্রপার্টি ইউজ করা হয়, জাস্ট স্কাল আর ব্রেইনের জন্য একটা হোমোজেনাইজড একটা প্রপার্টি, দ্যাট সর্ট অফ থিংস, এরকম একটা কি 2টা প্রপার্টি। এখানে কিন্তু মানে এক্সোনাল লেভেলের যেই প্রপার্টি গুলো সেগুলো ক্যাপচার করা হচ্ছে না, যার কারণে এটা ম্যাক্রো স্কেলে হয়তোবা ঠিক, কিন্তু রিয়েলিস্টিকলি ন্যানোস্কেল ইনজুরি ক্যাপচার করার জন্য এটা যথেষ্ট না। এখন কথা হচ্ছে বটম আপেরও আবার কিছু অসুবিধা আছে, সেটা হচ্ছে কনস্ট্রেন্টটা কিরকম, যে একটা ব্রেইনের মডেল আমরা এফইএম এ করলে সেটা 1 মিলিয়ন এলিমেন্ট দিয়ে করে ফেলতেই পারছি, কিন্তু যখন আমরা হচ্ছে বটম আপ করছি তখন 20 বিলিয়ন নিউরন, সেটার প্রত্যেকটার এক্সন, সেটার প্রত্যেকটা সাব এক্সোনাল কম্পোনেন্টের প্রপার্টি আমরা ইনকর্পোরেট করতে গেলে এটার যে মডেলটা হবে সেটা হবে আপ টু দিস ডেট এটা হচ্ছে ইম্পসিবল। মানে এটা এতই ডিটেইলড হবে। তারপরও হাইলি বায়োফিডেলিক মডেল তৈরি করার জন্য বেশ কয়েকটা রিসার্চ অর্গানাইজেশন এখনো চেষ্টা করে যাচ্ছে। সো আই থিঙ্ক দ্য সলিউশন ইজ এট দ্য মিডল, যে বটম আপ এবং হচ্ছে টপ ডাউন দুই রকম মডেলই হচ্ছে দুই দিক থেকে করে মাঝখানে একটা পয়েন্টে এসে মিলতে হয় এবং হচ্ছে একটা স্কেলিং ল অ্যাপ্লাই করে দুইটার মধ্যে কানেকশন ক্রিয়েট করতে হয়। সো ফার দ্যাটস মাই আন্ডারস্ট্যান্ডিং যে এই টাইপের প্রবলেম গুলো কিভাবে অ্যাড্রেস করতে হবে। আর ইউ পিজিএমডি অর অল অ্যাটম এমডি মডেল? আচ্ছা, সত্যজিতের কোশ্চেন বোধহয় এটা। এখানে হচ্ছে ও জিজ্ঞেস করতেছে যে অল অ্যাটম এমডি মডেল কি আমি ইউজ করতেছি নাকি কোর্স গ্রেইন অ্যাটমিক মডেল ইউজ করতেছি। আমার পিএইচডি ওয়ার্কে আমি আমার যেটা অ্যাড্রেস করা লেগেছিল সেটা হচ্ছে যে টাও প্রোটিনের মেকানিক্যাল প্রপার্টি, নিউরোফিলামেন্টের মেকানিক্যাল প্রপার্টি এবং মাইক্রোফিলামেন্টের মেকানিক্যাল প্রপার্টি, এটা আমাদের ক্ষেত্রে যেটা আমরা করেছিলাম সেটা হচ্ছে অল অ্যাটম মডেলই ছিল। কিন্তু এখানে আমি একটা জিনিস অ্যাড করতে চাই, সেটা হচ্ছে যখন মাইক্রোটিউবিউলের সাথে টাও প্রোটিনের ইন্টারাকশন আমার দেখা লেগেছিল তখন সিস্টেমটা হচ্ছে টু বিগ। কেমন? কারণ হচ্ছে সেটা আমরা যদি একটা ওয়াটার দিয়ে সলভেট করতে চাই সিস্টেমটাকে, এটা হচ্ছে মিলিয়ন অফ অ্যাটমস এক্সিড করে যাচ্ছে এবং মোস্ট অফ দ্য কম্পিউটেশনাল সুপার কম্পিউটার অ্যাক্সেস করে আমরা যেগুলো করতাম সেগুলো অনেক ক্ষেত্রে এটা অনেক চ্যালেঞ্জিং। সেক্ষেত্রে আমরা কিছু কিছু ক্ষেত্রে যেটা করেছিলাম সেটা হচ্ছে ইমপ্লিসিট সলভেশন ইউজ করেছিলাম, মানে অল অ্যাটম সিস্টেম বাট ইউ আর ক্রিয়েটিং অ্যান এনভায়রনমেন্ট সো দ্যাট as if the system is submerged in a continuum solvation environment. ঠিক আছে, এখন কোশ্চেন হচ্ছে যদি আমরা আরেকটু স্কেল আপ করতাম তখন কিন্তু হচ্ছে অল অ্যাটম সিমুলেশনে থাকা যেত না, তখন আমাদের কোর্স গ্রেইন মডেলিং এ আসলে যেতে হতো। সো এটা হচ্ছে একটা আমি বলবো যে রিজনেবল কম্পেনসেশন, যে সিস্টেমটা যখন আমাদের মানে সাব এক্সোনাল কম্পোনেন্টে থাকে, ইন্ডিভিজুয়াল সাব এক্সোনাল কম্পোনেন্ট টাও প্রোটিন বা মাইক্রোটিউবিউল এরকম, তখন আমরা অল অ্যাটম মলিকুলার ডাইনামিক সিমুলেশন করতে পারি এখন পর্যন্ত। কিন্তু তার চেয়ে যখন স্টেপ আপ করব, স্কেল আপ করব, মানে ধরা যাক যে একজন নেটওয়ার্ক বান্ডল অফ এক্সনস, তখন কি আর অল অ্যাটমে থাকা সম্ভব হবে না, কারণ হচ্ছে তখন সেটা 10-15 মিলিয়ন অ্যাটম বা তার চেয়েও বেশি হয়ে যাবে। তখন আমাদের আসলে কোর্স গ্রেইনিং করতে হবে, এটা হচ্ছে কথা, মানে অ্যাটমের সংখ্যাটা কমানোর জন্য।
Great. I guess Dada got his answer. If anyone else want to ask any question, I guess we are out of time. We have to end here. If anyone else wants to ask any questions or follow up, I think you can definitely reach out to Isaac Pai. He's available, I guess, through LinkedIn or whatever, or email address that would be more convenient.
Sure. Videoটা কি available থাকবে সেশনটা?
I think so. Whoever was recording, তোমাদের video কি record successful ছিল? Was there any problem? If anyone could unmute and Protoy, can you hear me? আচ্ছা, I guess বাংলাদেশ থেকে আর কি নেটওয়ার্কের সমস্যার কারণে আমরা ক্লিয়ারলি শুনতে পাচ্ছি না, but hopefully as far as I know ওরা হচ্ছে স্ক্রিন রেকর্ড করতেছিল, জুমের লগইন ইস্যুর কারণে জুম থেকে ডিরেক্টলি রেকর্ড করা যাচ্ছিল না। Hopefully within 1 or 2 days we will have a recording upload. What they usually do is upload the video in YouTube, so it's accessible to anyone anywhere. And usually they post it from the MMMRN Facebook page.
That's a good idea.
Protoy just confirm below the screen is recorded. Okay, so I guess we're good to go. We can stop here. We don't want to take any more time. Misabhai or anyone else, everyone else working. Okay, that concludes the presentation. Thanks everyone for joining today, and hopefully we will see you guys in the next webinar. Thanks. Take care.
Thanks.
Thank you,
Bye-bye.
Ishaq, for this, for your time. Thank you.
No problem. See you again.
See you, bye. See you, Dada.